Hello everyone, this is Medical Gateway, and today we are going to study a very important topic, which is cell organelles. Okay, so this is Guyton chapter number 2 part 3. Okay, cell organelles and their function, they are a complete story. Okay, so the story starts from the nucleus. Nucleus is basically the main controller of the cell. Why nucleus controls all the function of the cell? Because nucleus has a DNA, and DNA has a specific genetic material, a specific code. What they are coding? Basically, they are coding the protein, the formation of proteins. Now, DNA forms the RNA, and the RNA goes to the cytoplasm, and there the RNA on the ribosomes results in formation of proteins. Now, where are the ribosomes located? This is nucleus, and this structure is endoplasmic reticulum. This is the rough endoplasmic reticulum, and this is the smooth endoplasmic reticulum. Rough endoplasmic reticulum is said to be rough because it has a dotted appearance because of the presence of the ribosomes on its surface. Now, the transfer RNA transfers the genetic code to the and so the messenger RNA carries the genetic code from the nucleus to the ribosomes, and then the transfer RNA along with the messenger RNA and the ribosomal RNA all work together for the protein formation. Now, this protein can be structural protein formed on the ribosomes, or it can be functional protein. Now, functional protein are enzymes. Now, that's how the nucleus basically controls the function of all the parts of the cell. Number one, by providing the structure through structural protein, and then secondly, by controlling the functions by production of enzymes. Okay, now here on the ribosomes, which are located on the rough endoplasmic reticulum, protein is produced. This protein enters into the endoplasmic reticulum. Now, this protein will be processed in this rough endoplasmic reticulum, and then it will be moved out of the rough endoplasmic reticulum in these transport vesicles, and these transport vesicles will transport this protein to the Golgi apparatus, and in Golgi apparatus there will be post-transcriptional modification, post-translational modification. So the proteins are further modified in the Golgi apparatus, and then these proteins will be packaged in the form of these vesicles, which can be lysosomes or peroxisomes or different secretory vesicles. Okay, so this is the whole story of the cell organelles. Now the energy for all these functions is provided by mitochondria. So cell has a genetic makeup. It will define the production of proteins on the ribosomes. Ribosomes are located on the rough endoplasmic reticulum. Endoplasmic reticulum will modify the proteins by enzymes on their surface, and it will send the protein in the form of transport vesicles to Golgi apparatus, where the Golgi apparatus will do further modification, and then by budding off, and the packaging of the proteins will be done, and by budding off, the lysosomes, the different secretory vesicles, and the peroxisomes are formed. Okay, so this is the whole story, and the energy is provided by the mitochondria. So this is the whole story. Now let's discuss the whole story. This whole story is discussed in these two slides, where on the left side is the function of the nucleus, then the center is the endoplasmic reticulum functions, then we have Golgi apparatus functions. Okay, and then in the second slide we'll discuss the functions of lysosomes, peroxisomes, secretory vesicles, and mitochondria. So let's start the story. So the whole story starts from the nucleus. So nucleus is the control center of the cell. Why? Because nucleus has DNA, and DNA is deoxyribonucleic acid, and DNA has genes. The genetic makeup is in the form of the genes. Now, genes will define that which proteins will be produced, and proteins are of two types: the structural proteins and the functional proteins. The functional proteins are enzymes. That's how the nucleus basically controls the structure and the function of the cells by structural proteins and the functional proteins, which are enzymes. The genes also has genetic makeup for the reproduction of the cell by mitosis. Okay, now, so then let's discuss the structure of the nucleus. Nucleus has a nuclear membrane outside it. This is the nucleus. As you can see, there is a nuclear membrane outside. This is exactly the same lipid bilayer. Okay, the outer membrane is in connection with the endoplasmic reticulum. Here you can see this is a lipid bilayer. One layer is this, the other layer is this. This outer layer is in connection with endoplasmic reticulum. So here the endoplasmic reticulum starts. Okay, so the outer layer is in connection with the endoplasmic reticulum. Then there are nuclear pores in the nuclear membrane. So these are the nuclear pores in the nuclear membrane, from which the genetic material can pass out. The RNAs can move out. Okay, then we have nucleoli in the center. So this is the nucleoli. Nucleoli is made up of protein and RNA. So it's made up of protein and RNA. So it is a membranous structure. There is no membrane outside this nucleolus. Okay, and it is made up of protein and RNA. So one was DNA and the other one is RNA. DNA is deoxyribonucleic acid and RNA is ribonucleic acid. Okay. Now this RNA is present at two places. Now this RNA can be present in the nucleoli, and this RNA is also present in the ribosomes. Both the nucleoli and ribosomes are made up of RNA as well as protein. So nucleoli is made up of protein and RNA, and the ribosomes, which are present on the rough endoplasmic reticulum and also in the cytoplasm, they are also made up of protein and the same RNA. Okay, we will discuss it in detail later on. Okay, now then we have endoplasmic reticulum. Now endoplasmic reticulum is, as told previously, it is just located just outside the nucleus. Even the outer membrane of the nucleus is attached to the endoplasmic reticulum. And then the endoplasmic reticulum has these dots on it, these ribosomes on it. Okay, and then this is the Golgi apparatus and the Golgi vesicles, which are being budded off from the Golgi apparatus. Okay, now, so what is the function of endoplasmic reticulum? Number one, it helps process molecules. How it can help process molecules? Because it has protein enzymes in their walls. Okay, so it has protein enzymes in their walls which will help process the molecules. Then, as you know, the proteins are being produced on its surface. After the production, these proteins will be transported to the Golgi apparatus in the form of these transport vesicles. So this is the transport vesicle. The proteins produced on its surface will be transported to Golgi apparatus in the form of these transport vesicles. So the endoplasmic reticulum has its own lipid bilayer, just like the whole cell membrane, just like the nucleus. It has its own endoplasmic matrix. Okay, now then we come to the rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum is also known as granular endoplasmic reticulum. It has granular appearance because of the presence of ribosomes on it. Smooth endoplasmic reticulum is said to be agranular because it doesn't have ribosomes on its surface. Now, what is the function of rough endoplasmic reticulum? It's pretty simple. It has ribosomes on its surface, and it is involved in protein formation. And these ribosomes, the structure of the ribosome is made up of protein and RNA, just like the nucleoli. Nucleoli is made up of protein and RNA, and ribosomes are also made up of protein and RNA. Okay, so basically this nucleus has the genetic code. This genetic code will be transferred in the form of RNA. The messenger RNA will carry the message of the genetic code to the ribosomes. Okay, the code for the production of proteins, and there the transfer RNA will collect all the amino acids and required for the formation of proteins. So there are three types of RNA. One is messenger RNA, which will take the message from the nucleus to the ribosomes. The other one is the ribosomal RNA, by which the ribosomes are made. And the third one is the transfer RNA, which will transfer the amino acids, collect the amino acids from the whole of the cytoplasm and bring them to the ribosomes for the production of proteins. Okay, now, so the rough endoplasmic reticulum is, in short, responsible for the protein synthesis. Now, what is the function of smooth endoplasmic reticulum? Now, if proteins are being formed by rough endoplasmic reticulum, then it's really easy to remember that smooth endoplasmic reticulum is involved with lipids and glycogen. So smooth endoplasmic reticulum is involved with the lipid production and with the glycogen breakdown. Lipid production and glycogen breakdown. Now, smooth endoplasmic reticulum, when it produces lipid, that lipid enters into the walls of the smooth endoplasmic reticulum. And when it enters into the wall of the smooth endoplasmic reticulum, it helps in replenishing the wall, and it helps in the formation of transport vesicles. Now, as you can see here, these transport vesicles are being formed from the smooth endoplasmic reticulum. Now, as you know, these are formed by budding off. Now, these transport vesicles are basically formed of the same lipid bilayer. Now, lipid bilayer, as if you focus on its name, it is formed by lipid. So where is that lipid coming from? That lipid is being produced in the smooth endoplasmic reticulum that enters the walls, and those walls are being, when the budding off takes place, the transport vesicles are formed. Okay, so one function is this. Then the other function is that it has, smooth endoplasmic reticulum has enzymes which control the glycogen breakdown. Okay, and the third most important function of the smooth endoplasmic reticulum, which is asked oftenly, is detoxification. Detoxification of what? Detoxification of drugs. So it is commonly asked in MCQs that which organelle is involved in detoxification of drugs. Then you can say endoplasmic reticulum. So endoplasmic reticulum detoxify drugs by coagulation, by oxidation, by hydrolysis, and by conjugation. So how the endoplasmic reticulum detoxifies drugs? It detoxifies drugs by coagulation, oxidation, hydrolysis, and conjugation. Okay, these are the four mechanisms of detoxification of drugs by endoplasmic reticulum. Okay, so this was the function of endoplasmic reticulum. Now let's move on towards Golgi apparatus or Golgi bodies. Okay, now one function of Golgi apparatus is post-translational modification. So what is translation? Translation is the formation of the proteins. So when the proteins are formed on ribosomes and these proteins are being sent to the Golgi apparatus in the form of these transport vesicles, the Golgi apparatus will do post-translational modification of these proteins. Okay, so this is how it's done. This transport vesicle is then moved to Golgi apparatus. Their post-translational modification is done, and after its post-translational modification, a proper packaging of these proteins, these enzymes, as these secretions is done. Okay, So the vesicles produced by the Golgi apparatus has as the main as its if someone asks which organelle is involved in packaging of the substances, that you will answer it that Golgi apparatus is involved in packaging, and the packaging is done very in a very good manner. It has 4 or more layers of vesicular membrane. Okay, so packaging of what exactly? Packaging of the proteins, the enzymes. So after packaging, what the product is formed exactly? The lysosomes are formed. The secretory vesicles are formed. What are lysosomes? Lysosomes, we'll discuss it in next slide. Lysosomes are basically the main digestive organ of the cell. So lysosomes are being produced after the packaging from the Golgi apparatus, and then secretory vesicles are produced. For instance, any organ like pancreas. So pancreas will produce a lot of pancreatic secretions and pancreatic juices. So in the pancreatic cell, there will be a very huge amount of production of secretory vesicles from the Golgi apparatus. Then we have sweat glands. So all the glands or cells or organs which are associated with secretions like pancreas or sweat glands or apocrine glands, they will produce huge amounts of secretory vesicles by after packaging from the Golgi apparatus. Now, what exactly happens that these secretory vesicles, when formed in these secretory cells or organs, they have to be exocytosed. And how the exocytosis takes place? When the calcium moves in the cell, the exocytosis takes place. Okay, after the post-translational modification and the packaging and the release, what else? Do Golgi apparatus have some other function? Yes. Golgi apparatus also from synthesis of some carbohydrates. So which carbohydrates, to be precise? Hyaluronic acid and chondroitin sulfate. Okay, so this hyaluronic acid and chondroitin sulfate, they both collectively form the cartilage, the bone, the ground substance of extracellular matrix, and the mucus being produced by the glandular cells, or mucus produced in the glandular secretions. Okay, so Golgi apparatus is involved in the synthesis of carbohydrates. To be precise, which carbohydrates? The hyaluronic acid and chondroitin sulfate. So this hyaluronic acid and chondroitin sulfate are present in the bones, in the cartilage. They are present in the ECM is extracellular matrix, the ground substance around the cells. Okay, and these are also present in the mucus. Okay, and the last function of the Golgi apparatus is that its vesicles replenish cell membrane. When packaging is done and further vesicles are being released by Golgi apparatus, then these vesicles have to do exocytosis. When the vesicles do exocytosis, the vesicles does not completely move out of the cell. They just empty their secretions out of the cell, and the membrane of the vesicle become part of the membrane of the cell. Whenever the exocytosis is done, the whole vesicle doesn't move out. The vesicle is attached with the cell membrane. The secretions are being released outside the cell, and in most of the cases, the membrane of this vesicle become part of the cell membrane. That's how the Golgi apparatus replenishes the cell membrane. Okay, via its vesicles. Okay, so these are the functions of the nucleus, endoplasmic reticulum, and Golgi apparatus. Okay, so let's move forward. Now we have lysosomes, peroxisomes, and mitochondria in this slide. So lysosomes, extremely important lysosomes. Now lysosomes are formed by the Golgi apparatus. They are being separated from the Golgi apparatus by budding off. Okay. So lysosomes are known as the intracellular digestive system. Whatever is being digested inside the cell is digested with the help of lysosomes and their enzymes. So what exactly do they digest? Number one, lysosomes have enzymes which digest damaged cellular structures. Whenever a cellular structure is or cellular organelle is damaged due to any reason, it is being digested by the lysosomal enzymes. Then food, the most important thing, food is also digested by the lysosomal enzymes. And the third one is the foreign invaders like bacteria. They are also digested by the lysosomal enzymes. So these three functions are extremely important: the remodeling of the cells, the breakdown of the food particles to provide energy, and the killing of the foreign invaders. These are all high yield functions. So how the lysosomes kill bacteria? By lysozyme, by lysoferrin, and by maintaining an acidic environment, a pH of 5. Okay, so how do they break down a lot of these things? They break down with the help of enzymes. And what are the enzymes? The enzymes are hydrolases. Okay, hydrolases. Now if you focus on the word hydrolases, is the enzyme, and hydro is from H and OH. So hydrolases are the enzymes which perform breakdown with the help of water. Okay, so water is broken down into high H positive and OH ions. Now these H positive and OH ions will break the bonds of different things and perform the digestive functions. But there's one point you have to remember, that hydrolase always work in the acidic environment. The acidic environment activates these hydrolases. Okay, so which ones are the famous hydrolase enzymes? So we have lipase, protease, and nuclease. These all are hydrolase. Now lipase will break down lipids, protease will break down proteins, and nuclease will break down nucleus. Okay, so lipase, protease, and nuclease. Lipase for lipid breakdown, protease for protein breakdown, and nucleus for nucleus breakdown. Okay, so these are the functions of lysosomes, and these are the enzymes present in the lysosomes. Then we have peroxisomes. Peroxisomes are similar to lysosomes. Now let's discuss peroxisome. Now lysosomes were produced from by from Golgi apparatus, whereas peroxisomes are basically self-replicative. Okay. Then lysosomes perform breakdown by hydrolases, whereas peroxisomes, as their name says, oxysomes, oxys from oxidation, peroxisomes perform their breakdown function with the help of oxidation. Oxidation of what? Basically, the most important function is the detoxification of poisonous substances like alcohol. We also discussed the detoxification of drugs as a function of smooth endoplasmic reticulum. Here, the detoxification of poisonous substances is also done by peroxisomes, specifically the alcohol. If in the MCQ you are being asked that which organ performs the detoxification of alcohol, you have to write on peroxisomes. Then they also perform oxidation of long chain fatty acids. So they basically, peroxisomes catabolize long chain fatty acids. Now, which oxidases are present in peroxisomes? Now, this is H2O2 and catalase, hydrogen peroxide, the famous oxidases, and the catalase. They perform breakdown in peroxisomes. Okay, so lysosomes are produced from Golgi apparatus. Peroxisomes are produced by self-replication. Lysosomes perform breakdown by hydrolysis. Peroxisomes perform breakdown by oxidation. Hydrolases are basically lipase, protease, and nuclease, and oxidases are hydrogen peroxide and catalase. They break down food, damage cellular structures and bacteria, and they perform breakdown of long-chain fatty acids and detoxification of poisonous substances like alcohol. Then in the vesicles, we have secretory vesicles, as discussed previously in the case of pancreas. So secretory vesicles have secretory granules. Pancreatic acinar cells produce these secretory vesicles, which are in the form of proenzymes, and these proenzymes will get activated via different mechanisms. Okay, so these are lysosomes, peroxisomes, and secretory vesicles. Okay, these all are produced by Golgi apparatus. Now finally, the energy for all these processes is being provided by mitochondria. The mitochondria, the famous powerhouse of the cell. Why? Because it can produce ATP. Now mitochondria has two membranes, one this outer one, the outer membrane, and one this inner one which has folds in it, the inner membrane. Now this inner membrane, the folds in the inner membrane are known as crista. The significance of these inner folds, the cristas are that they have oxidative enzymes. These oxidative enzymes present in these folds basically perform the chemiosmotic formation of ATP, okay, which we'll discuss in a minute. Okay, now, so one thing is that mitochondria is the powerhouse of the cells. It has two membranes, and the inner membrane has oxidative enzymes. One other point you have to remember is that mitochondria is self-replicative. Mitochondria is self-replicative. These peroxisomes are also self-replicative. Okay. Now the ATP is produced by the chemiosmotic mechanism are used for mechanical work. The ATP is used to synthesize different chemical compounds. These ATP are used for transportation of different substances. So where the ATP is used for mechanical work, for synthesis of chemical compounds, for transportation of different substances. Now, how exactly the ATP is produced? ATP is produced by the chemiosmotic mechanism of ATP formation. Now, what exactly is this chemiosmotic mechanism? Chemiosmotic mechanism is basically that glucose, lipids, and amino acids, they all are converted into acetyl-CoA. This acetyl-CoA enters into citric acid cycle and Krebs cycle and is converted into hydrogen ion and carbon dioxide. Now this hydrogen ion, when combined with oxygen, will result into production of energy, which will be used by ATP synthetase, and ADP is converted into ATP. That's how the ATP is formed. Okay, now, let's simplify this. What we have to do, we have to produce hydrogen ion from glucose, lipids, and amino acid metabolism. If we can produce hydrogen ion from glucose, lipids, and amino acid metabolism, this hydrogen ion will react with oxygen and water is produced. And whenever the hydrogen ion reacts with oxygen to produce water, energy is produced, which will be packaged by ATP synthetase in the form of ATP. Now, hydrogen ion is produced basically when glucose, lipids, and amino acids are converted into acetyl-CoA, and the acetyl-CoA is entered into the citric acid cycle and the Krebs cycle. So this is chemiosmotic mechanism of ATP formation. Okay, by this we have finished the cell organelles. We have just discussed the points written in the Guyton. We haven't discussed extra points, or we haven't left any point. So this is it. These were cell organelles. See you in next part.